All right, welcome to the review video for the Human Microbiota lecture. I just want to walk you through what are the major ideas in this lecture and maybe show you kind of where to look in the slides and give you some examples of questions you might be asked. So to start, the first big topic is complexity. Why is it so hard to know what happens in our microbiota? Why is it so hard to know what to do to fix our health by changing the microbiota? I mean, what should we eat? What should we do? We don't know, and that's because of the complexity. And so if you go back to the slides, you look at all these numbers, and the numbers are kind of crazy. There's, hundreds of species, who knows how many different strains, and each one of them interacts with the immune system differently. There are higher level taxonomic groups that maybe we can learn something by looking at them, maybe not. But the basic idea is that we can learn what is in our microbiota. We can learn which genuses are there, which genera, sometimes which species, but we really need to know which strains are there and what's different between them. So that's the complexity that we need to learn.
One topic that wasn't originally in this review video that I have added is, where do the microbiota come from? What are the big drivers of which species are there and which ones are not there? And primarily I'm thinking about the colon, because that's where the majority of them are. And this is where you want to go back to your notes and look at the idea that the way a child is born is their initial exposure to microbiota. If it's a vaginal delivery, it's vaginal and fecal microbiota end up going into the neonate's mouth and colonizing the gut. Cesarean delivery results in skin bacteria going into the neonate's mouth and trying to colonize the gut, but not doing very well because skin bacteria are not really gut bacteria. And then the next big driver of what can be in the gut is breast milk, because breast milk is really specific in what kinds of nutrients are there, and there are a very small number of microorganisms that can actually digest them. And so microbes that can digest what's left over from breast milk in the colon will dominate the other ones in the colon. So as long as a child is getting breast milk, the microbes that can digest, like the breakdown products of breast milk, will completely dominate the microbial community in the gut. But then as soon as breast milk is taken away, then other bacteria have a much, much better chance of competing, and that's when you start to see a more adult-like microbiota. And from that point on, the adult microbiota starts to develop, and it's influenced by what bacteria the child is exposed to. So the bacteria the other people in the house have. And then eventually, once they're an adult, the main thing that's going to affect their microbiota more than anything else possibly could is what they eat, because that is going to determine what nutrients the microbiota get and what kind of chemicals they're competing for. So the drivers of that community change over time. And by the time you're an adult, you don't have much control. And really, the only thing you can do to shape your microbiota is change what you eat. Probiotics don't do much, in case you were wondering. Okay, back to old me.
Another thing we looked at is what controls it. So what is going to shape the microbiota? And early on, so somewhere in here, like this kind of stuff, early on, it's going to be shaped by what are the first microbes to get there. So during childbirth, what is the newborn exposed to? And then later on, it's controlled by the chemicals that, excuse me, the microbiota are exposed to. So look into that. Where is it? What are the sites where you'd expect to find the microbiota? And by far the most abundant is going to be the colon. So when I say the gut, I'm not talking about the stomach. I'm not talking about the small intestine. I'm talking about the colon. That's where the bacteria are, because feces is half bacteria by mass. If you look at the mass of feces, half of it is bacteria, very roughly. That's a huge number of bacterial cells. So that is by far the biggest site for our microbiota. After that, it's probably the oral microbiota and the vaginal microbiota. And then behind those, skin is where we have a lot of cells, but not nearly as much activity as we would see in the oral microbiota, the vaginal microbiota, or definitely the gut. So then, what effects does it have on us? So what are the big effects we know about? And there are really two big effects that you need to understand. One is colonization resistance. Okay, so what is that? That's, again, where the microbiota protects us from microorganisms that try to colonize us. So if a pathogen wants to grow in us or on us, sometimes our microbiota stops that from happening. That's colonization resistance. And there are several examples of that. What are some examples of colonization resistance? Some examples should be able to come up with are like C. diff, Clostridioides difficile. That pathogen typically doesn't grow in a healthy person, but if we disrupt a healthy person's colon microbiota, C. diff can grow there. And the strong evidence we have for that being colonization resistance is that when we disrupt the microbiota with antibiotics, suddenly something can colonize us that couldn't colonize us before. And so that's how we know. Another example is going to be sexually transmitted pathogens that the vaginal microbiota protects us against. And bacterial vaginosis is an example where that change to the microbiota removes its protective function of colonization resistance. The next big effect talked about is energy harvest. And this is where you can look into, volatile fatty acids. Yeah, volatile fatty acids. That's the main idea, where the colon is meant to be a bioreactor where we create this oxygen-free environment for obligate anaerobes to grow, and we use them to help digest the carbohydrates that we can't digest. And how that works is they just do what they normally do, and their waste products they generate, some of our cells can use them. So that's a story I might want you to tell. What are the parts of that? Under what conditions does it happen? Under what conditions might it not happen? And how is that oxygen-free environment maintained? It's a cool story to know.
And I'm just jumping in maybe 2 terms later to make sure you remember, what are the products those anaerobes make? What are the waste products that they release that have an effect on our bodies? are they used for? How are those waste products connected to maintaining that oxygen-free environment? So whether you think of them as a group of volatile fatty acids, or you can name butyric acid, propionic acid, acetic acid, those really are the products. And butyric acid in particular is the food for the colonocytes. Back to old me.
A really big idea is dysbiosis. This is where any change to the microbiota that leads to a loss of its benefit for us, to its loss of its function, that's dysbiosis. And that's the hot topic in the microbiota. The question is always, oh, if something's wrong with my gut bacteria, is that going to change some part of my health? If so, that would be dysbiosis. There are two parts to that definition. One is a change to the gut microbiota that is caused, and the second part is a loss of beneficial function. So make sure you know how to define that, and think of some examples of dysbiosis. Dysbiosis goes back to, in some cases, colonization resistance. So an example of colonization resistance, when we lose it, when antibiotics disrupt it and get rid of colonization resistance, that's dysbiosis. And bacterial vaginosis is a clear example of dysbiosis.
It sure is fun to listen to the version of me who was getting over a respiratory infection, and I don't know what it was. Maybe it was COVID. I don't know. But that's not current me. And current me wants to tell you that in fall of 2023 and for the foreseeable future, we are going to skip the mouse study and the intensive care unit study that previous students learned. But those are still in this video, so you can skip those. You don't need to study those studies.
Then we looked at two studies. So the mouse VRE study, make sure you understand that study, what they were trying to figure out. So they knew that the mouse normal microbiota would protect the mice from VRE. And that's colonization resistance. And they knew that because they knew if they feed the mice antibiotics and then immediately challenge it with VRE, the VRE can colonize because the antibiotics kind of clear the way for them. So what were they trying to learn beyond that? The human intensive care unit study, this is tricky because this is an example to me of a frustrating study that didn't teach us anything, but clearly shows us the challenge involved in learning about the human microbiota. That's why I show it, because it illustrates what we can't easily know. And the idea is they wanted to see if there are any patterns in fecal microbiota, because they could take fecal samples and guess what was in the colon based on the feces that comes out of the colon. Are there any patterns you can learn by figuring out which bacteria are in there? Could you look at the bacteria and predict someone's health from that? And all they could give us is phylum level identifications, like lots of Firmicutes bacteria, that's a phylum. Lots of Verrucomicrobia, that's a phylum. Proteobacteria, that's a phylum. And again, that doesn't get at the complexity of the microbiota. So there weren't any patterns. Even at that super zoomed out level, there are no patterns that would be predictive. And from there, we moved on to probiotic versus prebiotic. What's the difference? One of them can sometimes be a scam. One of them nobody even knows about, so they would have trouble scamming you if they're not trying to sell you something. And I I want to push people towards considering prebiotics because these are exactly what you need to get energy harvest to work. So if you want to start eating a lot of fiber and other things like that, you can make sure you have the right bacteria if you start eating prebiotics and feeding them. That's one idea people have. And then the last topic, really two topics, is the vaginal microbiota, which is complex, and it is shaped very strongly by the mucosa, by chemicals excreted by the mucosa for that purpose, both antibacterial chemicals, which would select for certain bacteria against others, and glycogen, which serves as a carbon source for bacteria that make lactic acid, and that lactic acid poisons other bacteria. That's a big function of the human microbiota in the vaginal mucosa. And then along with that, when that stops working, when there's a dysbiosis, that is bacterial vaginosis. And that's a condition where, for some reason, different bacteria are able to grow in place of the lactic acid bacteria. And so the pH of the vaginal fluid goes up by a point or so, and that lets other organisms grow that shouldn't grow, and that including sexually transmitted pathogens. And it also puts pregnancies at risk. And a large fraction of people who have this condition don't know it, because a large fraction of the cases are not symptomatic. And it's incredibly common. So for those reasons, everyone should know about it and understand it. It's hard to understand because no one knows how to cure it, and no one knows what causes it. That's the other wild thing. And scientists naturally gravitate towards any mystery like that, and it is a mystery. There are lots of risk factors for it, but no one thing that explains why it happens or doesn't happen. And no obvious cure. We don't know what causes it. We can't cure it. So it is a thing to learn about. All right, people. That is the human microbiota in a nutshell. That's the 15-minute review of the human microbiota. I'll see you at the next video.